Paddy Mallon, University College Dublin. I'm one of the VACCELERATE investigators, and I'll be moderating today's webinar. I'm delighted to introduce Professor Saul Faust from the University of Southampton. He's going to be giving our seminar today. Seminar is entitled Cov-Boost Safety and Immunogenicity of 7 COVID-19 Vaccines as a third dose booster following the Chad Oxford-1 vaccination, or the BNT162b2 in the UK. Just before introducing Saul, just a couple of quick housekeeping points, if I could. The first is that Professor Faust will speak for around 30 to 40 minutes, following which we would like to hold a question and answer session. I'd invite all participants to submit questions through the chat function at the bottom of the screen. I'll then collate the questions and put them to Saul at the end of the presentation. The VACCELERATE seminar series are CPD accredited. The VACCELERATE consortia have applied for accreditation from EACME for this seminar. We're just waiting on the final approval coming through. So for those of you who do require CPD points, Fiona and the team will be in contact after the seminar with details about how to acquire those points. So without further ado, I'd like to introduce Professor Faust. So Professor Faust is a professor of pediatric immunology and infectious diseases at the University of Southampton, and he's also the director of the Southampton NIHR Wellcome Trust Clinical Research Facility. He's a specialist in pediatric intensive care and infectious diseases, having completed training including an MRC clinical training fellowship, and subsequently underwent further postgraduate training, including a PhD in Imperial College London on the pathophysiology of coagulation abnormalities and meningococcal sepsis. He focuses on clinical trials in pediatric and adult infectious diseases, and has been involved in a number of sentinel vaccine trials since the onset of the COVID-19 pandemic, and has a very impressive research output just in the last 2 years alone to complement the pre-COVID research and clinical trials that he performed. So we're delighted that Paul has agreed to come and share some of these more recent data that will be very relevant to the VACCELERATE consortium, specifically in terms of establishing clinical trials and the approach to analysis of what is a very complex area. So Paul, I'll hand the floor to you, and thanks again for coming to speak to us today.
Thanks, Patrick. I'm not sure I enjoy long introductions, but thanks very much. So I'm going to talk about the Cov-Boost trial, which I was lucky enough to be handed to run by the NIHR consortium, more of which in a minute. Presume you can hear me and see the slides. And that's moved to my disclosure side. Patrick, do interrupt if you can't see me and the slides aren't moving.
All good.
I won't read out my disclosures, but I will just give you a second to look at them on the screen. So the Cov-Boost, the UK third dose booster trial, was one of a suite of trials that the National Immunisation Schedule Evaluation Consortium, chaired by Matthew Snape, was commissioned by the Department of Health and Vaccine Task Force in the UK to run. And this is an academic collaboration. The organisations involved are on the left-hand side. And Matthew, I believe, has presented to you before, or will be presenting to you, the heterologous prime boost trials, COMCOV, COMCOV2. There's also the paediatric trial, COMCOV3. Adam Finn and Rajeka Lazarus in Bristol led the flu and COVID vaccine trial, Paul Heath, PregCov. And I've led with my team in Southampton the Cov-Boost trial. And one of the things we've tried to do is keep laboratory assays standardized as much as we can across the NIHR vaccine task force trials. We've shared data managers and the electronic systems to make it easier for sites and participant diaries and training and so on. Together, we've enrolled about 5,500 participants. So the Cov-Boost trial that I'm going to tell you about was published, the day 28 data was published in the Lancet on the 2nd of December. It was a complicated trial. We had 7 vaccines to test because we those were the ones we'd been asked to test by the Vaccine Task Force in the UK. And we were also asked to test 3 of them at half dose. And so that was quite risky for us and for sites, both in terms of getting the trial recruited, because we were given very explicit instructions by VTF that we had to have the data back and presented to JCVI, the NITAG in the UK, at the beginning of September, end of August, beginning of September. So the lab work had to happen beginning of August. The 28-day bloods therefore had to be taken in July. Recruitment had to happen in June, and we were in April. So we didn't have very long, and there was quite a risk if one of the vaccines hadn't arrived, or on one hand, in IMP management, but also at the bedside, we thought 10 vaccines plus the control was too much for clinical risk of individual patient or research nurse basis. So we split the study into 3 groups, A, B, and C, and there were 2 or 3 COVID vaccines in each of those groups, and there were 6 sites running each of the groups, and each group had an independent meningococcal control vaccine. So you can see the design better here. So we recruited 2,886 participants, of whom about 25 in each of our subgroups were part of an immunology cohort as well. But in Group A, half the participants had Pfizer-BioNtech 2 doses, half had Oxford-AstraZeneca, and the vaccines were Novavax, half dose Novavax, Oxford-AstraZeneca, or control. And site Group B, half the participants again, Pfizer, half had AstraZeneca. The vaccines Pfizer, Valneva half dose, and Janssen. And similarly in Group C, the vaccines Moderna, CureVac, and Pfizer half dose. Pfizer half dose wasn't able to be in the same group as Pfizer full dose, because that was supposed to have been a different vaccine. So we had to switch it out at 2 days' notice with a substantial amendment for sites. So there wasn't the opportunity to change all the randomization schedules and drug supply to sites. So that was so when you look at the data, you can't look at Pfizer half dose and directly compare the stats with the full dose, although you'll see it doesn't really make much difference when it comes to clinical interpretation. Within each of these arms, 50% of the people were between 30 and 69 years of age, and 50% approximately were over 70 years and over. So I'm not going to talk you through all the baseline characteristics of all the 3 groups, but I'll give you an example from Group A. You can see here the interval between the first and second dose was variable. You can see about 70 days or thereabouts in the AstraZeneca prime boosted people, but in people who'd received BioNTech, this was mainly healthcare workers who'd received vaccine early in the program. So when we started off and Group A got going first, about a week ahead of everybody else, most of the participants were sort of 3 to 4 weeks between vaccines. That prolonged for Groups B and Group C. It came more to around the same 65, 70 days. And what was a weakness of our trial at the beginning, only 3 months, 10 to 12 weeks between the second and third doses, which of course was a bit higher in Group A because of this 3-week interval, and then switched to the UK to 12 weeks between the first 2 doses. But this weakness of the 3-month interval between dose 2 and dose 3 actually became a strength when the JCVI and UK government wanted to speed up the 3rd dose booster program with the advent of Omicron in November. So you can see in Group A, good balance between purple, under 70, ready orange, over 70, between male and female. Healthcare workers were a decent number, the purple. We didn't do so well at recruiting non-white people because of the rapidity and the speed with which we were trying to recruit, and simply there wasn't time to do extra work with those communities to get them on board. A range of baseline medical characteristics across the groups, and I'm not going to go through. And then finally, the age banding. 30 to 40, 40 to 50, 50 to 60, 60 to 70. From the bottom of each graph, the rainbow going up, with the biggest numerical group between 70 and 80, and then over 80 is in green. So you can see we've got good balance between the ages within our age cohorts as well. So I'm not going to go through the data for Group B and Group C. It's all in the Lancet paper if you want to have a look at it. So in summary, we were clearly looking for reactogenicity and immunogenicity, and the study was really powered to say, are there any vaccines or combinations that shouldn't be used? If, depending on vaccine supply in the autumn, what were the outliers, and should we be excluding things from the schedule and from the national program? So for reactogenicity, we saw any grade local and systemic reactions, similar after all vaccines. Fatigue and headache were the most common systemic reactions, pain most frequent local, and overall, as many trials have shown, reactogenicity greater in 30 to 69 years compared with older participants, regardless of the first vaccines they received. We did see that for people who had Pfizer as their first 2 doses, there were more local and systemic reactions after receiving Moderna, CureVac Generation 1, the AstraZeneca or Janssen vaccines as a 3rd dose compared to the other vaccines and controls. And also that people who received RNA or the Janssen vaccine after AstraZeneca had an increase in systemic and local adverse events. But overall, Moderna full dose, because we were using the full dose, because at that point, Moderna data on the full and half dose for the homologous schedules in the States had not been published or made public. We also found that Moderna full dose was extremely reactogenic as a 3rd dose, regardless of the first vaccine. We didn't see very much difference between Pfizer and half-dose Pfizer for pain. It was similar, but they were in different groups, so we could, statistically, that's not a valid comparison. And Janssen, Moderna, and CureVac also reported feverishness frequently, which wasn't seen amongst Pfizer or half-dose Pfizer reactogenicity. So I don't have to tell this audience about side effect grading, but we were really most interested in reporting to JCVI the moderate and severe, so mild, limitation of activity or marked limitation of activity, in order for people to the JCVI to decide on what should be the mass immunization program. And similarly, severe and moderate local grading, all detailed in the protocol. But these were the things that we're going to impact public health policy. So for the main study figure from the Lancet, these radial graphs show you the severe reactogenicity, severe local and solicited adverse events in the first 7 days. On the left-hand side, AstraZeneca, first 2 doses, and on the right, Pfizer with Group A, the top row, Group B, the middle row, and Group C, the bottom row. So you can see Chad after Pfizer was reactogenic, Janssen after Pfizer, but Moderna after both AstraZeneca or Pfizer, and that's across all age groups. I won't focus on CureVac, that having been Generation 1, having been discontinued by the company from further clinical development it was a 12 microgram dose of RNA that has been superseded now by GSK CureVac Generation 2 vaccine for development. So just looking in a bit more detail, and these are the figures from our supplementary file. So for Group A, this is moderate and severe combined, but split out into the ages. You can see that really the bottom left here, this is AstraZeneca after Pfizer in the younger people was particularly reactogenic, but really wasn't that reactogenic in the older people, and wasn't reactogenic after AstraZeneca. And similarly, Janssen, so an adenoviral vector after Pfizer, after RNA, but also after AstraZeneca, so adenovirus after adenovirus, top left, is reactogenic in the younger people. So the reactogenicity, if you like, is worse in younger people than older people. And then Moderna was just reactogenic in everybody across all age groups. So in the NIHR studies, we've been looking, like everybody, at the spike IgG titer, which has been done at the Nexcellis laboratories in Canada, which have got a validated assay, one of the CEPI laboratories, and we were asked to use the CEPI, the Nexcellis labs by Vaccine Task Force, who've overseen all these trials. And of course, we've also been doing the pseudo-neutralization assays, and we had aimed to do 4 variants when we set out, and viral neutralization assays in the immunology subgroups at Porton Down. So Porton Down were doing the VNAs, Bassam Hallis's lab, and PNAs were being done by Nexcellis in Canada. The delays in our data and our presentation of data have been entirely due to Nexcellis capacity and ability to give us data across all the vaccines, because it's actually quite hard to do a formal analysis or publish a paper until you receive all the data for a particular assay if you've been running a clinical trial that looks across multiple vaccines and multiple groups. So that sort of explains why things took a long time to report. We looked at across some of the correlations between our assays. We measured how the anti-spike IgG relates to the wild type PNA assay and found good correlation between anti-spike IgG and the wild type PNA. So you can see here for wild type, very good correlations. This is all vaccine groups. So I get AstraZeneca prime on the left, Pfizer prime on the right, group A, group B, group C, going down the 3 rows. Nice straight lines. So you can measure in a pre-Omicron era, a pre-Delta era, you could measure wild type antibody and know what would happen in the neutralization assay. Very good correlation. We also saw extremely good correlation to Delta PNAs. So this is just a little bit below, parallel, but below for all groups in the same fashion, good correlation. So if you measure antibody, you can also say what will happen to Delta. Very good correlation between Delta killing and the so if you make antibody, you'll kill Delta, not quite as well as you'll kill wild type, but perfectly adequately from a clinical perspective. And we showed the same correlation for the viral neutralization assay that's not shown in this presentation. So when we look at the spike IgG, we show the subgroup analysis by age for anti-spike IgG and cellular responses at 28 days after the 3rd dose between the study vaccines and controls. So we're looking separately at Chad primed and Pfizer primed. In each age group, looking at the geometric mean ratio compared to control in each group, adjusted for the things that you can read on the slide. So the pre-specified stats plan, remember this was April 2021, was to look for a minimum clinically important difference, anything anybody knew about what a 3rd dose would do, homologous or heterologous. And together with colleagues on JCVI and UK Health Security Agency and Department of Health, we chose a pre-specified 1.75 times control at day 28 as our statistical threshold. So the data, this is Chad, first 2 doses with the blue line in the middle of the picture. You can see the actual geometric means on the left-hand side column with controls in each group. To the right of this line, this is the GMR, favors vaccine, to the left favors control. So if we look first at the major RNA vaccines, Pfizer, Moderna, full dose Moderna and half dose Pfizer, you can see overlapping confidence intervals between Pfizer and half dose Pfizer and overlapping with the full dose Moderna. So not, from a clinical point of view, very good boost after adenoviral vector vaccine. We saw a surprising boost, 3.5 in the younger people, 3 in the older people, of Chad after Chad, a 3rd dose of adenoviral vector. We didn't expect that to do what it did. And an excellent boost, 5 and 6 times the control for the Janssen adenoviral vector vaccine as a second adenovirus after 2 doses of Chad. Novavax boosted well, Valneva boosted above our threshold after the Chad vaccine. CureVac did okay, actually, was the same as Janssen, but of course was only 12 micrograms and hasn't been developed further. After Pfizer, similar picture. The GMRs are less because the baselines are higher. So doesn't look as good, but it is as good when you look at the absolute values. So Pfizer, half dose Pfizer, full dose Moderna, actually all pretty similar. Again, Moderna, full dose Moderna, the strongest boost of them all. AstraZeneca after Pfizer, Janssen after Pfizer do just as well, especially in the older people. You can see the GMR, in terms of GMR boost, it's not as high in terms of the actual geometric mean itself, but in terms of the geometric, the GMR boost, the adenoviral vector after mRNA does very well. Valneva, on the other hand, the whole cell vaccine really did not boost at all after an mRNA first 2 doses, and CureVac was sort of equivalent to half dose Novavax at the dose that the company had chosen. Now, moving on to T cells, we don't, clearly we don't really understand the correlation between real world immunity and immunological memory and how that works, but we look across the Oxford-AstraZeneca assays that we were using against, and we think that T cells recognize more binding sites on the spike protein than the specific spike protein antigen that's seen by the antibody responses. So we think that T cells are going to be less susceptible to mutations in the spike protein, and maybe a boost and an ongoing presence of T cells across a broad range of variant responses gives us an indication of why it's possible that this is going to end up, down the line, as this is giving us an indication of why the real world effectiveness of these vaccines and immunological memory seems to be better than just the antibody responses, especially when it comes to looking at Omicron. So the trouble with the T cell assays is that the labs doing them globally haven't actually standardized between them yet, and the regulators are trying to get the labs to do that, of course. So there was no correlation between anti-spike and T cell assays at all over the place. So that doesn't help. Antibody doesn't tell us what the T cell responses are going to be like. So looking at the data in the same way, you can see not as pronounced differences between vaccines this time, but mRNA boosts T cells well to wild type after adenoviral vector first 2 doses. Janssen boosts extremely well, younger people, but also in older people, after in terms of the wild type T cell response. Not surprisingly, a 3rd dose of AstraZeneca didn't add anything. Remember, this was only 3 months after the 2nd dose. Novavax did all right. Novavax half dose, not so good. Valneva, not great, but it did boost. CureVac, again, dose problem probably. Looking after Pfizer, again, actually not so much variability between people who've had Chad and Pfizer with the 3rd doses, but all of the mRNA major vaccines boost, including the half dose and full dose Pfizer that look very similar. AstraZeneca after Pfizer does well. Janssen after Pfizer does well. Novavax does not so well after Pfizer compared to how it did against AstraZeneca. So Valneva didn't really boost at all, going along with alongside the antibody. Valneva doesn't boost well after mRNA in this trial. So just looking at some of the tables at the pan variant T cell responses, to get your eye into these tables: Chad on the left, Pfizer on the right, cellular responses, wild type, delta, and beta going down. So just picking out AstraZeneca after AstraZeneca, so not very much response, the GMR of 1.08 to get your eye in with a cellular response that clearly is doing something, because these people were not getting COVID, and at 3 doses they're still not getting COVID despite Omicron, believe it or not. The real world data is still holding up in the UK. We've got 40,000 people who've had Chad. They're the high risk people who've not been able to get to hospital or to another facility to be given RNA. and they're only a few percentage points behind the real world effectiveness for hospitalization and death. So the JCVI nor our DSMB have given these people an extra vaccine yet, and maybe that's because they, despite the lack of boost, they do have protective long-term responses. Come back to that maybe in discussion, talking about the importance of just whether we need to be chasing RNA in the future. On the other hand, if you look at Chad after Pfizer, you get this very good response, in the numbers in this assay up to about 100, and it's about the same. Wild type, delta, beta, there's no significant difference between those numbers, and a GMR of 2.5, 2.7, 2.9. Novavax also boosts really well after Chad. So you can see, hopefully you've got your eye in now. You can see we're in the hundreds across the variants after AstraZeneca with a good GMR, but actually in the 50s after Pfizer. GMR just under 2, not really meeting our criteria for cellular response Novavax after Pfizer. Moving to Group B, this is picking out Pfizer. Pfizer boosts well after AstraZeneca, and Pfizer doesn't boost quite as well as you might expect as a third dose at 3 months after itself on the T-cell response compared to how well it does after AstraZeneca. Janssen boosts very well after Chad or after Pfizer as across all variants. Valneva doesn't do very much at all as a third dose boost. Moderna, remember, really high responses after AstraZeneca, but at full dose, not the dose that's been used subsequently in policy. And full dose after Pfizer, again, very, very strong responses. CureVac did all right, but a dose response, as we've mentioned. So we've looked a bit at kinetics. So we looked at how quickly the antibody level reached the peak in a small group of people who had extra blood tests at day 7 and day 14, day 7, day 28 in this study. And you can see green day 7, blue day 28. So you can see for RNA vaccines, Pfizer, half dose Pfizer, Moderna, Chad on the left, Pfizer on the right. Maximum RNA values after third dose reached at day 7. And that, again, was really helpful to policy. So the study was able to help policy when Omicron came along and the program wanted to accelerate. We were able to say, first of all, that the third dose was safe and effective at 3 months, particularly safe at 3 months, because the original policy had been to have 6-month delay between the second and third dose, but it was able to be brought 3 months with the safety data from this trial, but also in public messaging, able to say that if you have a third dose of RNA, you will be protected in a week. It's worth having your vaccine with the Omicron wave coming, which was the public messaging. And didn't make any difference whether that was half dose Pfizer or full dose Pfizer. CureVac, you can see a stepwise rise up to day 28, again reflecting that maybe this is giving us a clue for the Pfizer and half dose, for the Pfizer and Moderna vaccines, that actually by the time you get to dose 3, the current standard doses are too high. We know that for Moderna full dose, they've already chosen to come down to 50 mics for subsequent doses. Maybe that needs to come down further. We'll come back to that later. And Pfizer half dose certainly seems to be as effective. Equivalent to full dose, half dose rather than half dose. So for global vaccine supply, that's really interesting. And CEPI have funded us to look at fractional doses, as they are funding other people around the world to look at fractional doses of RNA vaccines going forward. So we look, just looking at the other vaccines, stepwise rise for the adenoviral vector vaccines. You would expect to see that, of course, as they take longer to build immunity. Just looking at the difference between COVID naive and people who'd had COVID, we looked to see whether boosters were going to be worthwhile in people who had also had COVID. And you can see the red is still boosting in our mRNA vaccines, but also Janssen, and also also AstraZeneca after Pfizer. So COVID vaccination as a third dose is worthwhile whether or not you've had COVID as well as your first 2 doses of either AstraZeneca or Pfizer vaccines. So in summary of our data at day 28, according to our pre-specified criteria, all of the vaccines we tested boosted immunity after AstraZeneca, but only 6 of them after 2 doses of Pfizer. We have realized quite early on that dose optimization studies are going to be essential, especially with the RNA vaccines, maybe not so much with Novavax, which didn't do quite as well at half dose. I haven't really focused on that. And apart from the Moderna full dose, all the vaccines were acceptable side effect profiles, though actually even the Moderna full dose would have been all right if that had been necessary from an immunological point of view. So what are we still doing on the original cohort? Well, we originally planned to look at antibody and the immunological responses at 3 months and then 1 year. The 3-month anti-spike IgG have really suffered from Omicron, actually, because we can't just publish anti-spike. We need some viral neutralization or PNA assays. We're still 6 weeks away. The data that Nexelis received in October, the samples, we won't be getting the results for the PNA assays for at least another 4 to 6 weeks. So we are hopefully this week going to be receiving the last of the viral neutralization assays from Porton Down for the wild type, and we'll be able to submit the 3-month anti-spike IgG with some viral neutralization assay for all of our trial vaccines. Really, really interesting data that I'm afraid I can't show you, but what I can tell you is that by 3 months, there are different kinetics for different vaccine classes, and mRNA is not the only option if you want to look, or if your population it doesn't have a problem in the next 4 weeks. So with Omicron coming, we're thinking that antibody is really important to prevent transmission. That's in itself is perhaps a bit debatable in February 2022, but thinking let's get antibody as high as we can. Well, actually, by day 84, there are lots of different options to hit the same absolute level of antibody as RNA. So that is extremely interesting. And one of the things we've done as well is bring forward the 1 year sampling to 8 months. So everybody in the trial who's still in the original cohorts is going to have a blood test in February, which will be the last time point, because we want to investigate that kinetics going further. And hopefully the antibody data will come quicker out of the lab. The Nexelis labs are ramping up. By July, they will have hugely increased their capacity, but they're still not there yet. We're also looking at 1 and 3 month viral neutralization assays against Omicron. Again, suffering not being able to present this because of the lack of duplicates, because the labs were doing policy related questions with very specific groups of cohorts of people to provide data for policy, but that hopefully will be part of the publication. And we also, in October, the people who'd received the control meningococcal vaccine, because they couldn't be left unimmunized, and because Pfizer has become the sort of standard, we were able to decide that we would offer everybody on the in the control groups randomization to Pfizer, half Pfizer or half Moderna in October 2021. So they had a 6 month or 7 to 8 month actually delay between second and third dose. So we hope we have received yesterday actually the file from Nexelis for the antibody data, so that we can analyze the difference between a 3 month and a 7 to 8 month boost. Important for populations who haven't yet reached boosting with third doses in their population. So it will be interesting to see whether the kinetics of decline is different if you have a longer interval between second and third doses. We've had a few amendments to the study. We've got a late, so I've told you about the late, what we call the late boost study. That's the control groups. We also Paul Heath was CI of the Novavax trial in the United Kingdom, and there'd been a policy decision because you European colleagues, your NITAGs have not allowed the people on the trials to travel. Interesting discussion point we could come back to later on. But because of that, there was a decision by government to allow people who'd been in the Novavax trial to have extra vaccines in order to be able to travel. And it was noticed that Pfizer after Novavax was anecdotally giving a lot of reactogenicity. So we invited 111 people who'd received Novavax as their first 2 doses in the phase 3 trial to receive Pfizer as part of Cov-Boost to look at the reactogenicity and immunogenicity. I'm going to show you that data now. Actually, I was going to say in a minute. This is the data for Novavax, Pfizer. So it's not really relevant for European, US populations at the moment. But if any countries globally decide to deploy Novavax, then this might be relevant when it comes to thinking about what the 3rd dose might be. Very reactogenic vaccine. This is the local adverse effects. This wasn't actually a hospitalization. This was misclassified by the participant on their diary. This should have been read severe. So not hospitalized, but very painful, very red for quite some time. Also systemic adverse events, really quite bad. This was, if you like, the most reactogenic schedule. We've tried to visualize this using the radar graph. This is the Lancet Group A graph I showed you before, and you can see this is just the severe, but this would have been the most severe. It would have been probably just a bit worse than Moderna. I can't say that, of course, because it's not statistically comparable, but it looks bad even compared to the Moderna full dose that we'd seen originally, and that's the severe and moderate together, and that looks bad as well compared to what we saw before. So Pfizer, after 2 doses of Novavax, is a really reactogenic schedule that people will need to bear in mind later down the track. We don't yet have the immunogenicity data yet from this schedule. This is unpublished, so please don't talk to this data outside of this group. Pfizer and Novavax have seen that data, of course. So in terms of our CEPI-funded fractional dose study, we're trying to answer the question, can fractional doses of approved mRNA vaccines produce as good responses in 18 to 30-year-olds who's previously received 2 doses of Pfizer or Moderna on the UK schedule as a full dose of Pfizer in people who have and haven't had COVID. So our study vaccines for a study that's recruiting at the moment are Pfizer, 1/3 Pfizer, so the pediatric dose, the 5 to 11 pediatric dose, half dose Moderna and a 1/4 dose Moderna, 18 to 30s, and we're hoping to recruit about 465 COVID naive, 496 people who've had previous COVID, about a 1/4 of the way through recruitment at the moment. Recruitment in this age group is slow in the UK, but so is the national programme in this age group. So we're going to pursue that till mid-March at least. We were also asked by Vaccine Task Force to look at 4th doses in people who'd received Pfizer and Pfizer. So we've got 2 questions. Can Pfizer, given as a 4th dose, reach the same IgG spike at day 14 after the 4th dose as it did after a 3rd dose? And the same question for Moderna at half dose. And you'll see we've switched to day 14 primary outcome in order to try and get things sped up to the lab and back from the lab, because we think, well, we know we get maximum doses at day 7 for the 3rd dose. Population is people who had Pfizer last June, regardless of their first 2 vaccines. We actually recruited 80% of our original cohort, so 160 people, which was, we thought, pretty good. Data should be back we've got the spike. We were expecting the spike assay back. Actually, this got prioritized, so we would expect the IgG data in the next week or 2, we hope. And finally, we were asked by VTF to recruit to an Moderna Omicron vaccine sub-study to determine if neutralizing antibodies against Omicron is superior after the 1st dose of the Moderna Omicron compared with the 4th dose of Pfizer. Also, so we're looking at neutralizing antibodies against Delta and whether they're non-inferior after the 4th dose. Complicated, because the only way of doing these studies is viral neutralization against Omicron and Delta. So is wild type relevant anymore? Probably not. So we've powered this on doing all of the recruited participants. It's about 200 in each group. All of the recruited participants into full viral neutralization assays for Omicron and Delta. And we think this is even more important as data not my data, but as data is reported, that the spike antibody and Omicron viral neutralization don't correlate very well in some studies. Though I haven't got that data to show you and haven't actually clapped eyes on it myself. So just in way of summary, the policy decisions supported by this study were that Pfizer and half-dose Moderna were used as the vaccines for the UK third dose booster program. Heterologous schedules are safe and immunogenic as a third dose. We use a 3-month interval to speed up the program. 7-day maximum antibody response communication. WHO guidelines have quoted the Cov-Boost data, and ongoing, we're tracking longevity of immune responses, and I've given you a hint that the kinetics look to be very different, and we will get some indication in humans of a 4th dose response, both the wild type and Omicron variant vaccines, as the head-to-head heterologous schedule first look. Very finally, Patrick, if I can, just a nod to our amazing regulator and health research authority and ethics committees in the UK. The MHRA have adhered to every single standard that they had before and maintain after, but their openness to discuss with us has just been amazing. So we could design a sub-study, email them, within a couple of hours they'll email back and say, yeah, that looks good, or actually should we have a meeting, how about Friday afternoon, and we'll get together and we'll meet with their statisticians, and they'll advise what they think and what they will need in order to be able to use the data. So they've maintained their independence completely. They've maintained, we have to make the study decisions, but this new relationship we've got with the regulator to be able to rapidly design and deliver important trials, I think is something that's going to last into non-COVID work. And just one thought for you, is the future fast enough? This group, this community of people are critical if we're going to develop a new global regulatory approach. And somehow it feels as if our American colleagues, and apologies if any of you are, have just let us down here. The FDA, by their decision making across the pandemic, therapeutics, vaccines, rigid, still not regulated, an inability to communicate, no ability to think about heterologous schedules in the same way. Somehow as a community, we've got to work with our American colleagues to actually bring them to the table to let us do heterologous schedules from the beginning of vaccine development. Because I think what these data, what the COMCOV data have shown you, is that homologous vaccines aren't necessarily the way you might want to deploy them in real world. And unless we do those heterologous trials from the beginning alongside the homologous ones, things are just going to take too long. We know, because we work with them, and we still work with them, that the manufacturers are willing to engage, but they're paralyzed. They can't do anything, and they can't change the regulatory advice. We're going to have to do that as an academic community. So I'll leave you there. Thank everybody who took part in massive study team. And sorry if I've gone on a bit longer than I was supposed to, but hopefully generated some good questions, good discussion.
Saul, thanks a million. That was a real tour de force of vaccine research, and I guess one of the things that strikes me about the UK's approach in general is that when you look at the likes of MHRA, how it's worked with NIHR in terms of the national funder for clinical research, and then the distributed the distributed collaborative nature of what's going on in the UK. I think it really stands out as a lesson for the rest of the world in how you can prepare better at a national level to ensure that research feeds into policy rapidly. So definitely congratulations to you and your team for having produced research that's impacted policy at such a number of levels. There are a few questions. I suppose that the big thing that's been touched on by a couple of people is the kinetics, and as you know, as you've already said, you can't comment too much on that. The fact that you brought forward the 12-month to the 7-month window, is that a bit of insight into maybe how rapidly declining some of these kinetics may be?
No, we want to see if it's real, because if it's real, mRNA is not the only game in town, and I guess that you know, it could have just been a blip, and I think for global policy, you know, it's quite exciting. I think I really worry that, you know, because of the way the media have is it controlled the pandemic, driven the pandemic. We've done things in policy that people might not have wanted to do again. You know, thinking about the dose decisions around 12 to, somebody's talked about 12 to 15-year-olds. Matthew Snape's doing those studies, and he is planning on doing a reduced dose, third dose study as part of COMCOV3. That's being discussed at the moment, is the answer to that question. But you know, that some of the decisions we've made, like going with maximum dose for children in 12 to 15 before the data was really there as to whether that or a 10 mic dose was better. I don't think, you know, I'll go back. I don't mind being directly critical of the FDA. I think they've done global science a massive disservice in the way that they've been handling this, and you know, thank goodness for the 3 mic decision with pediatrics, you know, to delay that until they've actually got the data, because you know, as a pediatrician, I'm sitting here thinking, well, you know, colleagues are, I'm not on JCVI, but I know that JCVI have been really careful in thinking, you know, should you immunize children? Where do you go with pediatrics when they've all had COVID and they've had Omicron? Do we even need to give them a vaccine when it's not that we don't want to immunize children. It's not that I'm anti-vax. I'd immunize my children if I was offered it. But we've still got too many people in Europe, the States, and everywhere who haven't had their first 2 doses, and we've still got people globally who've not, you know, who are vulnerable, who haven't had the 2 doses and a 3rd dose. That's where we should be concentrating on our resources, not on people who don't need it, or where it, you know, it's a designer vaccine for travel. You know, what we've got this very odd situation in the UK today, where there's a recommendation apparently coming that 5 to 11 immunization is going to be voluntary, but all that's going to do is the white middle classes who don't need the vaccine are going to get it for travel, because countries and politics has driven, you have to have vaccine X or vaccine Y to travel. You know, where's the French decision that you have to have a 3rd dose that's RNA come from? That's not science-based. Who made that? Sorry if any of you were involved in that decision, but where's that come from? It's totally insane. Omicron's been and gone. You know, there's no suggestion that you have to have an RNA vaccine as a 3rd dose. So I just, somehow as scientists, we've got to bring back get the right questions answered and try and influence our nutty politicians, and they're nutty everywhere. The UK politicians are nuttier than most, let's face it.
Yeah, I think it's, you know, I've often grappled with this. You know, in infectious diseases, you would think to yourself, there's no point in immunizing someone who's already immune. And I suppose the challenge with COVID-19 has been defining what would be a threshold of immunity beyond which you wouldn't vaccinate. And you know, I think one of the things that I noted from your data that we're struggling with in VACCELERATE is this concept of, you know, the primary endpoint for a vaccine study. And, you know, within Cov-Boost, you went for the 1.75 fold over baseline, but you can even see within that it really depends what your baseline is.
Yeah.
If you get an mRNA baseline, you don't boost as well, but your absolute antibody levels, because they're so much higher. Do you get a sense from the graphs that you showed and from the data that you showed in terms of the correlations between antibody levels and the PNA and VNA? Is there a threshold even for the wild type or for any of the others that you think it provides some sort of correlative immunity?
No, well, we weren't powered for correlative immunity, and I think Omicron's been a game changer with that. I mean, I think some of the data that VTF colleagues have told us about in the last week, when we've been thinking about, we've been thinking, do we really need to wait for these VNAs or the peer? Could we do it on something on PNA? The answer is that we're going to have to do the full VNAs for the Omicron assays, because the game has changed. We've been, I think the best answer to your question, Patrick, is we've been really, really worried about our group of people, half of whom are over 70, who've received Chad, or let's face it, on our data, Chad, half Novavax. So our over 70s, those two groups, so our CureVac and our Valneva, they got taken out the study and given Pfizer with these, before Christmas last year, because the CureVac would have been fine as a booster, but it's discontinued, never going to have a chance of approval, so they had to be given an approved vaccine. Valneva really hadn't worked, so those people who received Valneva were also offered Pfizer as a fourth dose, off trial, but as an NHS vaccine. So the groups that we were left worrying about are Chad and Chad Novavax, and the DSMB have been, hounding me to hound Mary Ramsey and UKHSA colleagues for that data. And even last week, it's good news. It's really good news. I think Nick, there's a New England, UKHSA have got a New England paper coming out, but even the latest data suggests that Chad, and this is people who received this as the high-risk group last summer, so June in our study, but September in the national program. These people are not at more risk of hospitalization and death. So it goes back to that point I made in the talk. We've lost control of transmission control with vaccine. There is no, we don't have to be as mad as my government and get rid of testing or get rid of isolation and, just pretend that, did you all know, for example, that COVID's no longer a thing on the 31st of March in the UK? It's officially ended, research funding's ending, and influenza's coming back into our hospitals. That we've gone completely mad on one respect, but we've lost control of transmission globally because Omicron is different. It doesn't. On the other hand, we're doing brilliantly with hospitalization and death. And if we can get 2 doses into everybody globally and a 3rd dose into everybody who's vulnerable or over 70, we've got a really good chance of the world going back to normal. And that means not wasting resources on children or vaccines on children, not really bothering about the 20 to 30-year-olds, because although they're good to study and we'd like to know, just in case the pandemic changes again and a more lethal variant comes along, we have to, that's the only reason I can think, so to go back to your correlation of protection, well, hopefully we'll get to a stage where we're just boosting the most vulnerable once a year, but everybody's paranoid at the moment.
Yeah. But just the data on the Novavax mRNA was really interesting. And I just saw a comment from Martina in the chat, Martina Sester, that Germany's currently recommending this. So your clinical experience, you'd be a bit cautious about that sort of approach, just from a vulnerability perspective, or?
So is this for people who? I'm really interested in what Martina said, because we didn't think there was a rush to get these data out. Right, we thought it's okay. Novavax only just got approved. It's going to take months for this to be a problem, because you can have Novavax. It's only the people on the trial where this was a problem, because it hasn't been deployed yet in the population. So we've got a few months before anybody's going to be getting RNA in a national program, haven't we? Or have I? Is that wrong?
It looks here as if it's a current recommendation. I'm not sure if maybe Oliver or Martina could clarify that. There's Martina's just said we're just waiting for the MVX vaccine to come. So at the moment it's recommended until more data are there.
Yeah. So we've got some time, haven't we, to get this data out to look at. We haven't got the immunodata back yet. We haven't got the antibody yet. So again, that should be in the next few weeks.
Okay, so but really timely then, I think.
But it is, it's important, particularly for the LMICs, because this is a it's a vaccine that's easy to transport, that has been purchased by domains, and actually it's important to find out whether we could use, I don't know, say Janssen or Astra instead. And as I said, it might well be that we only looked at Chad, Pfizer with a third dose. But I think if our 3-month data is borne out by the 7-month data, then it really is important that we stop just focusing on the absolute antibody levels provided by mRNA and actually say, what is the long-term protection offered by different combinations, and does it actually matter what you get? And maybe heterologous is actually what you need.
So this leads me on to another point. I know we've only got a few minutes left, but there were 3 things I wanted to cover with you quickly. Okay, so you've got the antibody levels, you've got the T cell responses. I suppose the big thing, especially as you start moving into third doses and maybe beyond, is the concept of the memory B cells and how important they are in this whole thing. Any plans within Cov-Boost?
Plans, yes. This is really interesting. Seppi suggested we look at this too. So there is a pilot project going on actually in Matthew's team and Andy's team in Oxford. So we're all collaborating. They're looking at some of the ComCov data to see whether they can use the frozen PBMCs to look at the B cells, because I think you're right. We sort of know what T cells are going to do now. We can, to a certain extent, we can stop doing that many assays. Interesting to look at Omicron, but you're right. Haven't seen any of the preliminary data, but we are hoping to do that, Patrick. Yeah.
And obviously that you did highlight that the whole issue with doing any of this type of research, especially when you start looking at functional assays, is you've got the harmonization, but you've also got lab capacity when it comes to doing, especially the live viral neutralization assays. It's something in VACCELERATE that we're also struggling with. What do you see there being an answer from either a European, a regional, global perspective in terms of trying to address that for the longer term?
I mean, somehow we all have to do this under, working with, I know, say BASAM is working with Nexelis, is also working with CEPI. Communication and prioritization, I think, and avoiding competition. I think this is a massive problem. We know they're building a new lab at Portland. They're building, Nexelis have just been taken over by, is it Q Squared and IQVIA? There'll be expansion there. Other people have got labs. I think this is where the regulator becomes so important as part of the conversation, because it's no point in doing policy trials if the policy and the regulation in the countries isn't going to align. So we had to be, and when we've gone back, we've been a bit anxious about the Omicron VNA for the Omicron variant vaccine. VTF said, look, you're going to have to go back to the MHRA and just point out that this BASAM's assay is not a validated assay. The only one that's validated is wild type, So we went back to the MHRA and said, just to remind you, that's not validated. Is that okay? And they gave us a beautiful answer. It just said, look, for your first look at policy, that's fine. But if it's going to go any further than that into documentation, then it's going to need to be backed up with the validation that'll need to be X, Y, and Z. So we're okay to start the trial and proceed and get the assays going, but at some point, BASAM's going to have to do that validation, and that's going to get even more complicated if we start having bivalent vaccines, not just variant vaccines. So your point, Patrick, is absolutely critical. There's no point in doing this in isolation. Everybody's got to use standardized assays and share capacity and sort of plan capacity at a global level somehow.
I know that the EU have a couple of them. They have these important projects of common economic interest. It's like this funding vehicle, and I think that they're trying to use that at a regional level to build up capacity, within that. But all of these things take time, and as you rightly point out, it only takes a couple of weeks for a variant like Omicron to come along, and a lot of these assays end up being nearly redundant.
And useless. I mean, we cancelled. We had contracted with Nexelis to give us alpha, beta, delta, and wild type, and they only got halfway through the alpha and beta. They were being so slow. We ended up having to just say, look, forget it. Don't even complete it. Nobody's interested in Cov-Boost Day 28 alpha and beta anymore. It's intellectually, it's worthless for policy. It's worthless for intellectual output. Do something else better with your time.
We've got another question here then from Mary Jason Lang around that, the relationship that you have with MHRA, and you talked about how responsive they were, the face-to-face discussions.
Yeah.
How did that work in terms of it? Was it informal, or did everything end up being?
No, formal minuted meetings. So we wrote to them with a question. They either wrote back, yeah, that's fine, or the meetings we respond. Fair enough to them. We had to minute the meeting and send them the minute to say, yeah, that's okay, because to scribe it was hard, and we could allocate somebody to do that. But no, it's all formally documented and in writing. And I know actually MHRA team themselves are organising a series of international meetings with their colleagues, because I've been asked to speak at one in a couple of months' time, to say how we worked with them. So I think our problem is not Europe, because I think that European colleagues, everybody can see the value. You can have a conversation like this, and people will come to table, and MHRA and EMA used to work together, right? They were the same people until halfway through the pandemic. So our problem is the FDA.
The concept of equality for people that participate in vaccine trials in terms of their, the access to travel freedoms that the general public get. I was on a call earlier on this afternoon that Andy Ustinowski was involved in with the WHO. They're trying to, I suppose, push through an initiative to address this at a global level. It goes back to what you said about a lot of vaccination is driven by nearly the need to travel, and it's highlighted again and again that people are disadvantaged being on trials and not getting that access. What are your thoughts there in terms of what we should be doing? Maybe some suggestions for VACCELERATE as a consortium.
Yeah, well, we've been fighting this since March 2021. Patrick, it's really, really, really critical, because the public are going to stop taking part in trials. It's creating such bad feeling publicly. Our previous Secretary of State, Matt Hancock, took it to G7, got agreement with everybody except one European country. I won't tell you who that was. We know in retrospect, but 6 out of 7 premiers agreed it. Whenever the G7 was in Cornwall, it was blocked by one European politician, and then it didn't happen. Chris Whitty, who's the UK Chief Medical Officer, who was an infection epidemiologist, argued this with ministers in the UK publicly, with investigators present. He said, look, on an individual level, vaccine trial participants are A, low risk, even if they've received placebo, because they're going to be taking precautions and wearing masks, and B, minuscule compared to the number of people who are going to slip through the net, regardless of whether they've had vaccine or not. And it was his opinion that regardless, if you're on an approved vaccine trial in your country, whether it's EMA or MHRA, FDA regulated, that could be agreed, then it doesn't matter which group or which phase of vaccine, you should be able to have full global travel exemption, and thanked for your part in keeping the world safe. For some reason, politicians, probably Brexit, probably because everybody hates Britain now and doesn't want to take any notice of pretty much everything, which is fine because it's a stupid thing to have done. But for global vaccine policy, for people taking part in clinical trials, for VACCELERATE, you need your countries to do the same, or people won't take part in trials. Because if you're on a trial, you can't get a certificate.
Yeah, and I think that's one of the learnings that we've certainly taken on board. And I know at the EU level in VACCELERATE, we've had quite a good EU response in terms of getting access to the IMP for trials. Oh. And I think we need to leverage the same pathway.
And the EU wasn't the problem at G7. The EU were fully supportive, from what Chris Whitty told us. That wasn't, and even Matt Hancock does. It was a one individual country that then took the momentum away. And again, unhelpful United States in all this. But it's politicians who need to believe in this. If they want the world to be prepared, they have to be prepared to support the work that you and we do.
And I think if there's any example of how important research is in terms of driving policy, Cov-Boost is that example. And I think, as you say, the policy implications are global. We have gone over time. I just want to thank you again for the really fascinating insight and also the fantastic discussion after. And,
sorry about the politics.
No, but I think it's science and politics with COVID seems to be the way things may be of the future in terms of pandemic responsiveness. But yeah, thank you for your time. And we'd like to thank everyone for joining and participating and asking questions, and thank the VACCELERATE team in the University of Cologne, Oliver's team, for hosting this webinar. And look forward to meeting everybody again at the next webinar.
Yeah, thanks a lot.